Thank you for joining us today at ParkOn. It's a real honour to be with you today. I'm Amelia, I'm Research Participation Lead at Parkinson's UK, and I'll be facilitating this session about what new treatments are currently in development. The idea of this round is to leave you feeling hopeful and positive about the interesting things that are coming up on the horizon. And I'm very, very honoured to have three brilliant experts today coming to talk and share their points of view about these things. The idea of this session is it is an amuse-bouche, a taster of these items. All of the things that will be discussed, there will be links to, so don't worry, you don't have to scribble down like fury. But at the same time, don't be surprised if you end up with a lot of questions out of these. Both Simon and Kevin are going to be talking to you today. I'm sure could talk for many, many hours about each of these topics. And what we wanted to do is give them an opportunity to almost elevator pitch them to you. So you could then come into our live Q&A session that will happen after this and ask lots of questions of them, as they're very excited to do that. Now, I'm going to introduce you to our three panelists. Firstly, I'd like to introduce you to Paula Scurfield. Paula is here as a person with Parkinson's who's got a real interest in research and has been involved for many, many years. Paula was actually the fourth person in her family diagnosed with Parkinson's. Her passion for research to find a cure started when watching how difficult life became for her parents and her late father, who was diagnosed in his 80s. At the moment, halting or slowing down the progression of Parkinson's seems unobtainable, and this can make people with Parkinson's feel out of control of their bodies, and Paula really experiences this. And so by participating in clinical trials and other research initiatives, she feels like she's doing something useful, and it's very empowering. So I'm very pleased to invite Paula to the panel today, as she will share her thoughts and reflections on what our other two panelists will be talking about. I would also like to introduce Dr. Kevin McFarthing, who has a PhD in biochemistry. His career includes senior R&D positions in life science research products with Amersham, diagnostics with Serrano, and consumer healthcare with Rickett Beckinser. So has a lot of experience in industry. He also ran his own consultancy business focused on the effectiveness and efficiency of innovation management, which I'm personally struggling to get my head around, but I'm sure you can ask all sorts of questions in that direction. Kevin was also diagnosed with Parkinson's in 2012, so it comes from both sides of the fence. Since then, he has actively engaged with research into the condition. He is joint editor of the Clinical Trials Highlights section of the Journal of Parkinson's Disease and a member of the Research Committee of Cure Parkinson's. He also collates the Parkinson's Hope List, which I'm sure will be mentioned and we'll hear a bit more about, and there will be a link to at the end. And then our third panelist today, definitely not least, but unfortunately last because of the alphabet. Simon is the Deputy Director of Research at Cure Parkinson's Trust, obviously working with Kevin. The Trust is an international support of both lab and clinical-based research of Parkinson's. Simon's originally from New Zealand, and so has come a long way to join us over on this side of the planet, and has over 15 years of experience in the field of Parkinson's research in both academic and the biotech sectors, so has a great wealth of knowledge. He's been involved in lab-based research as well as clinical studies with a number of scientific publications, so you can definitely look up Simon's name afterwards and I'm sure you'll find lots to read. He also maintains the Science of Parkinson's website, which attempts to explain in plain English the research currently being conducted on Parkinson's. So as you can see, we have an absolute wealth of knowledge on the panel today, and I'm very, very excited to invite them all to share everything with you. So today, to break it down into what we actually think would be an interesting thing for you to learn about and what's coming up in the future, we decided to break it into two sections. So the first section is about novel treatments or novel drug treatments, and then the second section is non-drug treatments. So I would like to invite to the table first Kevin to talk to us about his novel treatment that he's chosen. And Kevin's going to talk to us about a novel drug called Anavex 273. So can I pass it over to you, Kevin?
Thank you very much, Amelia. Yes, as you said, Anavex 273 is the subject, my first subject. Anavex is the name of the company, as well as the part of the name of the drug. And what is it? Well, it's an activator of the sigma-1 receptor. And so the next question, obviously, is what is the sigma-1 receptor? Well, it's a chaperone. It helps to fold proteins. And it's in a part of the cell called the endoplasmic reticulum. When that gets stressed, then proteins can be misfolded, can pile up and jam the roads, if you like. And then with the help of chaperone proteins, they can not only get rid of the roadblock, but also try to help restore normal protein folding and normal protein production. The danger is if that isn't done, then more stress can happen and eventually lead to cell death. So it's important to find compounds that interact with the sigma-1 receptor to prevent the accumulation, to restore normal protein production. It's also influencing other pathways, and so net it can promote plasticity of synapses, which means that they can adapt and regrow. It influences the survival of neurons by helping to release BDNF, brain-derived neurotrophic factor, which itself promotes the survival and growth of cells. Now, Anavex 273, also known as blarcamesine, doesn't trip off the tongue lightly, has been tested already for Alzheimer's disease and for Rett syndrome, RETT syndrome. That's a very rare postnatal genetic defect which affects almost exclusively young girls and has severe motor problems and cognitive difficulties. It's quite a rare disease. About 1 in 10,000 live births will develop Rett syndrome. And they've come up with very interesting results in Alzheimer's and Rett syndrome on the treatment of the disease-associated dementia. And what they've done is also done a phase 2 study in Parkinson's disease dementia. As most of you probably know, the risk of somebody with Parkinson's developing Parkinson's dementia is relatively high. So it's a very high priority area for researchers to look at for novel treatments to deal with that condition. So what Anavex have done is they've done a phase 2 multi-center randomized double-blind placebo-controlled parallel group 3-arm 14-week study in people with PDD, and there were 132 people involved, testing 2 doses and a placebo. So a screening and an observation period, and then a 14-week treatment period, and followed up with just a safety check. They've also added another study, which is taking the people who took part in the original trial into an open label extension to look at safety. What they found was quite amazing, really. In terms of cognition, the primary testing was the cognitive drug research computerized assessment system continuity of attention, of which I know relatively little, but it's a standard tool used to assess cognition in longitudinal studies. And they found a significant improvement, quite a large improvement in the cognition after 14 weeks. Now, a secondary outcome measure was the MDS-UPDRS, the Unified Parkinson's Disease Rating Scale. In the placebo group, that went up by an average of 3.5 points over the 14 weeks. In the high-dose treatment group with Anavex 273, it went down 11 points, a gap of 14.5 points between the active and the placebo. And that was only after 14 weeks of treatment. So it's extremely fast. We can only speculate what's going on, apart from the fact that the level of messenger RNA for the sigma-1 receptor rose in the serum and was positively correlated with the level of function of each patient. So Is it disaggregating alpha-synuclein fibrils? Is it restoring a loss of function that the aggregation of alpha-synuclein deprives the cell of? We don't know. But we also have to have a little bit of caution because the results haven't been published in a peer-reviewed journal yet. It's just got a company announcement. They're consulting with the FDA for a phase 2/3 study, which may be able to be classed as a pivotal study for registration. So exciting, but cautious optimism, I would say, is the name of the game for Annex 273.
Thank you so much, Kevin. That was really, really interesting. And I'm sure Paula's been scribbling away like fury. So now I would like to pass us over to Simon. And Simon is going to give us his elevator pitch of his novel treatment area is the results of the Denali phase 1 study for their LRRK2 inhibitor and other recent developments in this area. So over to you.
Thank you very much, and thank you very much, everybody, for coming and watching us today. So in almost every cell of our body, we've got DNA, and it's a long string of genetic code, and it is interspersed by sections of DNA called genes, and these are the functional regions. These are the regions that provide the instructions for making proteins. And there's about 20 to 30,000 of these genes across a stretch of DNA. A single strand of DNA. And over the last 25 years, what researchers have noticed is that tiny little errors in certain regions, certain DNA, regions of DNA, these genes, are associated with an increased risk of Parkinson's. And one of the most common of these, what we call genetic variants, genetic variations, is in one of the genes called LRRK2. This is leucine-rich repeat kinase 2, or LRRK2. And what happens is when you have a genetic variant in LRRK2, the protein that is produced quite often becomes very hyperactive. And in the carefully balanced environment of a cell, a hyperactive protein is not a good thing. And it can lead to stress within the cell and ultimately cell death. Now, what's interesting here is that people with LRRK2 variants have this hyperactive LRRK2 in their cells. But what researchers have also noticed is that individuals without the LRRK2 variant, so people with what we call idiopathic Parkinson's or just spontaneous Parkinson's, also have hyperactive LRRK2 in many cases. And so this has led researchers and biotech companies to develop inhibitors of LRRK2, little small molecules that will bind onto this hyperactive LRRK2 and calm it down or stop it completely. And one of the biotech companies that's been leading the charge in this area is a company called Denali Therapeutics. And in May of this year, they released their phase 1 clinical trial results for the first LRRK2 inhibitor that's been in clinical trial. And the results are interesting. The study was made up of 2 parts. Firstly, a single and multi ascending dose study in healthy volunteers for up to 28 days. And then there was a second part, which was 3 different doses of their drug, which is called DNL151, 3 different doses of it in people with Parkinson's over the span of 28 days. Both studies found that the drug was safe and very well tolerated, and no adverse events or bad stuff was observed. And when they looked at LRRK2 measures in the blood, they found that in all of the doses, they saw a reduction of at least 50% activity. So that's encouraging. And they also looked at other biomarkers, which also indicated that the molecule was engaging with its target and having a positive effect. Now, the studies were too short to say whether it was doing anything with regards to slowing the progression of Parkinson's, but the results are encouraging enough that a large pharmaceutical company called Biogen has stepped in and partnered with Denali Therapeutics to take this molecule forward into phase 2 and 3 testing. And the companies are now organizing 2 large late-stage clinical trials. One will be in people with Parkinson's who have the LRRK2 genetic variant. And then there'll be a second study, which will be in people with Parkinson's that have spontaneous or idiopathic Parkinson's. So this is something that will be starting next year, this late-stage study that will be determining efficacy of the actual drug. Now, in addition to all of this, because this is an exciting target for Parkinson's research, there are a number of other biotech companies and pharmaceutical companies that are exploring this area. GlaxoSmithKline, the large pharmaceutical company, is partnered with 23andMe, and they have a LRRK2 inhibitor program. The pharmaceutical company Merck has a LRRK2 program. There's a biotech company called Scurfield Therapeutics. They have a LRRK2 program, and there's also another company called Escape Bio. There's a lot of companies quietly working away on LRRK2 inhibition, and I think this will be an area to keep an eye on in the coming years.
Brilliant, thank you. That is a whistle-stop tour and actually really interesting area of research. So thank you so much for that. Paula, over to you. What are your thoughts on what you've just heard about? I know it's a lot, but I gather you've been scribbling away there. You just have to pop yourself off mute, Paula.
Yes, I've been scribbling away. I have to say, these 2 guys are so brainy. They make my brain hurt just listening to them. But I truly feel quite inspired and hopeful after listening to both of them. One thing that probably scares me more than any other symptom is losing my cognition, losing my marbles. I didn't have that many to start with. Any drug that's going to enable me to keep what marbles I have is to be applauded. I suppose one of the things I've learned through having Parkinson's and being involved in research is what we all really want is something that's going to cure our condition. But second best would be some sort of modification to our disease, something that would slow it down, if not halt it. And both of these drugs that Simon and Kevin have introduced offer hope in that area. I suppose one of the things I would be wondering is at what stage would participants need to be placed on the drug, but that's something I can perhaps find out later. I always think it's a good idea if we can capture patients at an early stage, not that people like me and Kevin don't matter because we've had the disease 7 years and 9 years respectively, but any treatments that can get to people early is good. I do really want to focus on this selection criteria. So often when you see calls for research participants, they're saying somebody diagnosed in the last 2 years, somebody diagnosed in the last 5 years. And if you think that from the time we actually see the consultant to be diagnosed, we may well have had the symptoms 1 or 2 years. And the fact that our brains have been deteriorating for 10 or 20 years before that, I think this issue of the stage that we get patients involved in these drug trials is really, really important. Focusing particularly now on Simon's drug, I understand that approximately 10% of people with Parkinson's have a genetic link, genetic mutation underlying their condition. And I do know that these patients have a particularly quick progression, and that can actually be quite young. They're very often at working age. So if you think about the benefits to society of a drug like Simon outlined, we're going to enable people with Parkinson's to be working longer, paying their taxes, paying their national insurance, supporting their families, let alone the emotional consequences, which would be huge, wouldn't they, to people to be able to continue working. I know a GP, I worked with a GP who had to give up medicine because of his Parkinson's. And he told me, and it breaks my heart every time I think about it, that not a day passes when he doesn't miss medicine. I have a very close friend who's a teacher who had to give up teaching because her balance was so bad. She kept dropping all her textbooks and the kids would scurry around picking everything up. So any drug that would help people with a genetic mutation has to be applauded. I was particularly interested when he said that those of us with idiopathic spontaneous Parkinson's also have a hyperactive LRRK2 mutation. So it's not just the 10% of people with the early onset with the genetic mutation, it's the rest of us, it's the Kevins and the Paulas. And I'm stunned by the number of companies and very, very much cheered by the number of companies that are getting involved in the LRRK2 modification. Because obviously these drug companies have to make money, and there are, in fact, diseases which are much more widespread and financially rewarding for drug companies. Anyway, guys, you've cheered my day up considerably. Thank you very much for your marvellous presentations.
Thank you, Paula. And that was just the first lot. So let's trot on into the second lot, where we're going to talk about the non-drug treatments. So Simon, I'd like you to elevate pitch first. And your area of interest you'd like to introduce us to is Blue Rock Therapeutics and upcoming cell transplantation clinical trials for Parkinson's.
Yes, so Blue Rock Therapeutics is a biotech company that is very actively involved with cell transplantation for Parkinson's. Cell transplantation represents one potential avenue for therapy with Parkinson's. Parkinson's is a neurodegenerative condition, so that means that we're losing cells in the brain. And one way of treating Parkinson's is to go back and replace some of those lost cells. Now, that this was experimentally attempted back in the 80s and 90s with clinical trials focused around fetal neural tissue. It was controversial for the ethical reasons, and there was a very limited supply of the actual cell tissue that was to be transplanted. So what over the last couple of decades, researchers have worked on developing protocols from taking embryonic stem cells. So these are cells that are capable of becoming anything at all, and gradually encouraging those cells to become firstly neurons and then dopamine neurons, which are the cells of interest in Parkinson's. They are the cells that we are predominantly lost in the brain or predominantly affected by Parkinson's. Now, Blue Rock Therapeutics is one company that has developed a special protocol for making these dopamine neurons from embryonic stem cells. And on the 8th of June of this year, they announced they had transplanted their first patient. So this is a phase 1 study testing the safety and tolerability of the treatment. And it will involve 10 people. It's only a very small study initially. And they are being transplanted at research sites in the US and Canada. They will be followed for 2 years. And the results of the study should be available in 2024. Now, this is an exciting development in a very active field of clinical investigation. We currently here in the UK have the TransEuro study, which is involving the old method of cell transplantation, but it's developing tools around that can be taken forward for the new procedures. So better assessment, better surgical procedure. And those techniques will be applied to a new study that is going to be kicking off in 2022, which is called STEM-PD. The STEM-PD is taking embryonic stem cells, growing them in cell culture, encouraging them to become dopamine neurons and transplanting them into the brain. This is will be a collaboration between researchers at Cambridge University, also at Lund University, and a pharmaceutical company called Novo Nordisk. So it's going to be a big study that'll be kicking off next year. Professor Roger Barker at Cambridge is one of the leads of that research. Then there's also the Kyoto study, which is being conducted present in Japan. And this involves a special kind of embryonic stem cells. They've taken skin cells, and through a magical trick of modern biology, what you can do is turn that skin cell, you can take it back a step in its development and turn it back into an embryonic stem cell. So you've gone from a skin cell to an embryonic stem cell, and then you can encourage it to become a neuron and a dopamine neuron. And the researchers in Kyoto, Japan, have been transplanting a number of individuals. I think we're up to about 3 or 4 at present, and they've been following them over time, making sure that the treatment is safe and well tolerated. But a pharmaceutical company called Sumitomo Nippon Pharma is stepping in and expressing interest and hoping to take this procedure forward for larger clinical trials in Japan and also in the US. And then finally, there's a company called Aspen Neuroscience, another small biotech company. This is in California. And they have also developed a stem cell protocol for turning stem cells into dopamine neurons. And they are hoping to start clinical trials very shortly as well. There's a number of other research groups. It's a very active field, as I say. And I think we will be seeing a lot of research coming out of it in coming years. So it's a very exciting area.
Brilliant. Thank you, Simon. That is yet again another whirlwind of all sorts of exciting stuff that's going on. And I'm very, very happy to pass the floor over to one last time to Kevin, who is actually going to take us on another different, should we say, route, is looking at adaptive deep brain stimulation.
Thanks, Amelia. DBS, deep brain stimulation, first started in the early 70s. And since then, at least 175,000 people around the world have had a DBS device installed. That's just from one company, Medtronic. There's obviously many more companies playing in the field, and so the number will be even higher. So it's an established technique. It's also been improving in different parameters in recent years. So the hardware developments have improved, so directional leads, helping to stimulate specific targets, sensing electrodes, new programming, new software to help the areas of the brain respond to different settings. So there's more anatomically accurate programming and the potential for closed loop feedback. More of that in a moment. DBS is also starting to be managed remotely by telemedicine. The accuracy of the placement of the electrode has also improved. And now it's quite common for people to be asleep during the DBS procedure. So at the moment, they're what's called unidirectional. You adjust the setting on the controller to a level that gives the best outcome for the patient. It's a bit of a trial and error process, and obviously subjective. It's also dependent on the clinician's interpretation as well. So imagine a situation where you could have the device itself measuring things locally and responding from those signals, making its own mind up to respond in the most appropriate way to what's happening in a relatively short time frame and on the micro scale. Imagine if you had a bike with 7 gears and riding in gear 3 was the best for you. But in order to change gear, you had to get somebody else's intervention. That would not be the best way to ride a bike. If you were able to respond to the steepness of the hill, going up or down, you'd change gear accordingly. It's maybe a crude analogy, but it's the best one I could think of to try and look at the difference between open loop DBS and closed loop DBS. There have been various clinical studies done on closed-loop DBS. And in one study, with the electrodes placed in the subthalamic nucleus of the brain, the normal DBS reduced the scores in the UPDRS by 30%. The closed loop reduced the UPDRS scores by 50%. Also, because the closed loop system isn't working every single microsecond, it's not always on. It's on when it needs to be, and it's on at the right level when it needs to be. There was a 44% reduction in the power needed from the battery. The battery replacement is a challenge, relatively improving challenge these days, to replace the battery when it runs out. It's another surgical intervention. Now, there's also some work starting on the next generation of adaptive DBS and closed-loop DBS, based on the local detection of neurotransmitters. So if the electrode was also able to measure the dopamine in the striatum, and if there's not enough dopamine there, it would give more of an electrical boost, and that again might lead to a much greater element of control and stability within DBS. There's still plenty of issues to overcome, so measuring neurotransmitters with electrodes inside the brain is not a trivial achievement. Also, some of the electrodes that are used to measure can undergo what's called biofouling, with different cells attaching to it and growing on it. But there's a clinical trials about to start with a device called the Percept from Medtronic, another one called from Alpha DBS, and it's clinical trials like that which hopefully will improve the performance of DBS, the reliability, the lifetime of the devices, and everything else by using adaptive DBS.
That was really, really interesting and so much stuff covered. Paula, I suppose we come to you now and ask you, for your initial reactions and thoughts on what Simon and Kevin have just shared.
Well, thanks, Amelia. I now feel I've been hit on the head by a very large cricket bat. It was an interesting comparison that Simon and Kevin made between starting off with conservative treatments and then going on to invasive treatments. My great aunt, in fact, was one of the very, very first patients ever to have DBS. She had quite the worst tremor of anybody I've ever met. And so somebody operated on her, first one side, then the other. And it made absolutely no difference. So we've come a long way since. One of the things that's always slightly worried me about having traditional DBS is the fact that we have to undergo, we used to have to undergo stereotactic surgery. So we were kept awake and the brain was placed in a sort of metal harness. But everybody I've spoken to who's had DBS has benefited from it. And I think it very much depends on what stage you are at the disease, what you're prepared to go through, doesn't it? Sounds a bit scary to me, but I've never known anybody have adverse effects, and everybody I know has had benefits. The DBS that Kevin was talking about is, I think, known as fourth generation, is it not? There's been quite a lot of changes to DBS over the years. And I was talking to somebody earlier today who's been following the Movement Disorder Society Congress that's going on at the moment. And she mentioned something called transcranial magnetic stimulation, which is similar to DBS, but it's a non-invasive procedure. But it stimulates externally the same area of the brain. Now that's my kind of surgery. But I do like the idea that the patient himself or herself doesn't have to have something in their hand to adjust the stimulation. I think that's a huge improvement. I think that's all I have to say on that. Moving on to the stem cell transplants. There are ethical issues, I think, with stem cell planting. We were living in the United States at the time under a Republican government, and I think it was George Bush Jr. actually stopped the research on stem cell transplants because they are very much influenced by the religious right, and people saw ethical constraints, that there are ethical constraints to manipulating stem cells, which of course are the most basic cells that we will start off with, and then they're modified over time. Once that's fair enough, the theory, but once you have a condition, I think your thinking very much changes. And I have to say, I have absolutely no ethical constraints about having stem cell transplant. I'm interested in the stem cell, the pluripotent stem cells that where researchers take the skin of the patient and then in a Petri dish manipulate the 5 proteins to create a stem cell, to turn it into a neuron, basically. I think that's incredibly exciting. Again, it is invasive, but I'd be prepared to go ahead with that if my symptoms got bad enough, and presumably one day they will. The only other concern I have about stem cell transplants is it is incredibly expensive. And I think for Japan, which is really a world leader in the pluripotent stem cell arena, Japan is a fairly wealthy country. But I wonder whether it might not be a procedure that's too expensive for some of the less industrialized countries. I'm going to finish in a second, but I did meet the professor who got the Nobel Prize for Science for his work on pluripotent stem cells, and got the opportunity to talk to him. And my understanding is the Japanese government have passed a law where only one side of the patient's brain can be transplanted. So it is quite heavily regulated by government, which is good. These things need to be regulated. But again, chaps, incredibly exciting for those of us who have to put up with some of the rather mundane but irritating and embarrassing symptoms of Parkinson's. So thank you.
Thank you so much, Paula, for those really, really interesting thoughts. And yet again, thank you to all of you for, sharing your knowledge, teaching us these introductions to all of these different exciting things. There was a lot covered, and I really hope it has triggered some of you in the audience to think about things to do with you and what you're interested in. This is deliberately designed for you to go away and ask more questions. As I said, there is a live Q&A session after this where you can ask those questions to Kevin, Simon, and Paula. I hope you were typing away in the question box during this. All of the things that we've been talking about today, as I mentioned at the beginning, the links will be available. Also, other links that we're going to make available to you are for those of you who are interested in potentially finding out more and finding active research trials. As mentioned, these research trials that we've talked about today, they're not recruiting as it stands at the moment. A lot of them are coming along in the future. Not all research recruits in the UK, but we do try and share all the opportunities that are going on. If you'd like to find out what research is taking part that we know about that is actually looking for recruits right now, please go to our Take Part Hub, which is on our website. It's under the research area, and the link to that will be available after this. Also, as mentioned at the beginning, Kevin's Hope List, the resource that has all of the research that's coming up and going on, that will be a link to that, so you can go and have a look and a read at that. in your own time. There is a lot in there, just to warn, but it's very exciting. Also, if you are someone who's interested in sort of research that's going on further afield, there are a couple of different places you can go and have a look, which is the Fox Trial Finder, which is Michael J. Fox, which is linked up with a lot of US trials, as some of them were alluded to earlier by Simon, and also trials across in Europe. Also, wider than that in the UK, there is the National Institute of Health Research Be Part of Research Gateway. So if you don't have Parkinson's, or if you are someone who is a friend or a partner and you'd like to take part in research and you see what else is going on there, do go and have a look there. Finally, if you would like research opportunities emailed to you that we will know about, then join our RSN if you're not already a member. The Research Support Network is an amazing resource, and we do make sure we keep you updated, not only with opportunities to take part, but where you can receive more information, read our blogs, also have your say in our research and the research that we know about to really shape the future, and how important the user journey is within research coming up. So that's all from us, and we really look forward to receiving your questions in the live Q&A. So thank you again to the panelists, and thank you again for your time and input, and looking forward to your questions. So welcome to our live Q&A session, and I'm really honoured to have Simon and Paula here. Unfortunately, Kevin can't join us today, but I have some fantastic questions that you guys have submitted to us via Slido that I am going to bounce to Simon and Paula, and I'm sure they will bounce them between them as well, because we're that kind of group of people. So the first question that I have that's at the top of our Slido options today is, how do you know if you have a genetic mutation or a LRRK2 mutation. And so what I think I'd like to do is just ask Simon that first, but then come to you, Paula, with that same question, because I know you've had your genetics tested. So Simon, can I...
So the only way you can find out if you have a genetic risk factor for Parkinson's is to actually have your DNA sequenced or analyzed. And this typically involves either a blood test or you spit into a test tube and you send that away for analysis. And the scientists will break open the cells in the sample and they'll take out your DNA and then they'll put it through a machine that just goes through and reads off all the ACTGs. And they line them up, put them in order, and then they go looking for these tiny little variations, these tiny little differences between your DNA and a very wide sample of DNAs. And DNA is made up of regions that we call genes, and these genes provide the instructions for making a particular protein. And sometimes within some of those genes, there are mistakes. You have 3 billion of these T's and G's. So there is always going to be one or two mistakes. Each of us has these variations. It's what makes us each unique from each other. But some of these variations in some of these genes are associated with an increased risk of developing Parkinson's. It doesn't mean you're going to get Parkinson's. It just means that you have a higher risk of developing it. For example, LRRK2. There are variations in the LRRK2 gene that mean you have a higher risk of developing Parkinson's. But you can only learn this by actually having your DNA sequenced.
Thank you so much, Simon. So yeah, I'm going to come to you, Paula, because you've had your DNA sequenced. Could you tell me what that was like?
Yeah, it was actually very easy. The reason I had it, I had my whole genome sequenced was because I'm the fourth person in my family to have Parkinson's disease. So I was referred to a David Cameron initiative, which was called the 100,000 Genomes Project. And the aim was to map the genome of people with rare and complex diseases. And in fact, I don't have a genetic mutation for Parkinson's. My Parkinson's is idiopathic. But giving the blood was easy. Several members of my family also gave blood. And of course, the result was negative for all of us.
Thank you so much. And in terms of, I suppose, sort of the aftercare and all that kind of support, do your family still talk about it?
Yeah, well, we talked about it it took an awfully long time to get the results. I mean, years rather than months, because I believe the blood samples were sent off to Denmark. And then it all came back and everything had to be analysed. We don't talk about it now. One of the reasons I was interested in getting tested and having my son tested is that my son and his partner had a baby a couple of years ago, and we just wondered whether there would be a genetic risk both to my son and to the baby. But they now stand the same chance of getting Parkinson's as anybody else on the planet.
That's really interesting. And I think, those family conversations and kind of keeping things going, it's really, really important as we're, understanding genes. More and more people are having their genes sequenced. So it's a good conversation, I think, to start having with families sooner rather than later, for sure.
Yeah, I think could I just say something, Amelia? Sorry to interrupt you. One of the things that is important if people have any kind of genetic testing is that they should be counselled. Because if you know that you have a genetic susceptibility, which is all it is, a genetic susceptibility to, say, breast cancer or Parkinson's or any other disease, it can be a bit like the sword of Damocles hanging over your head. So it's important people are aware of that. And I will just mention, I am taking part in a study at the moment. I think it's called Frontline. It's the principal investigator is Professor Anthony Shapiro at the Royal Free Hospital. So there is always research about genetics going on, and that is a way for people to both participate in a trial and help future generations and find out whether their Parkinson's is genetically linked.
Yeah, absolutely. And there's lots of research, and actually the more research we see coming out, I think genetics are definitely starting to be more commonly talked about and included within a lot of research trials. So thank you. The next question I have on my list is actually about deep brain stimulation. So the person who asked this question said that they personally had deep brain stimulation 15 years ago, and that obviously things have moved on since then. And he was asleep while the electrodes were inserted. How far do you think deep brain stimulation can take us? Simon, can I give that one to you?
So the big exciting area within deep brain stimulation at the moment is what's called adaptive deep brain stimulation. That's where, because typically with deep brain stimulation, you have the electrodes implanted into a particular region of the brain, and then the technicians will calibrate the stimulation and have it at a particular setting. And that's what that particular setting is, what you have, and you go about your business, your daily life. And sometimes it needs to be the volume turned up or the volume turned down, but it's pretty much a standard protocol to keep you active and mobile. With adaptive deep brain stimulation, what you have is a recording from the brain communicating to the stimulator. And so it's adjusting moment by moment to give you the best setting. So that's a very exciting area of research at the moment, adaptive deep brain stimulation. And there's clinical trials ongoing for that presently.
Fab, thank you. And Paula, have you ever thought about or had conversations about DBS?
I've had conversations about it. I'm 7 years into my disease, and at the moment I'd frankly rather jump off a cliff than have neurosurgery. But if my symptoms become bad enough, never say never. The fact that there is the opportunity to have stem cell transplant or DBS is something I'd never rule out.
There's a lot of other options now coming online as well. There's a lot of preclinical experimental work, but there's also entering the clinic what we call high-frequency ultrasound, which is a non-invasive approach, but it just targets a particular region of the brain and zaps it with a bit of ultrasound. And it's a very straightforward procedure to have. You go to the clinic in the morning, you walk out half an hour later. There's no stitches, there's no surgery. The only issue there is that there is no volume control. You can't program the intensity of your response to it. Whereas with DBS, you can program and continue to program going forward. So that's one of the considerations to take into account.
Do you know, that follows wonderfully into another question that we've actually been asked about repetitive transcranial magnetic stimulation, or RTMS, and kind of what do you think of that as a therapy for speech and motor improvements?
It's still pretty experimental, is I think the easiest answer. There's data to suggest that it is having an impact, and there's data to suggest that it's not having an enormous impact. The issue with it is the targeting. I think quite often they have a paddle that they just apply to a region of the head and see what the effect is. I'm oversimplifying it there, but there needs to be refinement and more research is probably the appropriate answer.
I think that's definitely more than an appropriate answer, in the fact that actually with all of these things, and actually a lot of what you guys all talked about and shared, is that this is an ongoing process. There's a lot of research that needs to be done in all of this. And I know, Paula, you've been involved with research for quite a long while, that actually seeing things sort of come through is really interesting. And actually, I've got a question from another person here. Is there any evidence yet that current treatments influence the course of the disease? And is there an indication that novel drugs will achieve this? So I'm going to ask Paula, what's your perspective on that first? Just give Simon a little bit of a break, if that's okay.
Oh, I was truly hoping you were going to ask Simon that question first. Would you like to repeat the question? Because I have Parkinson's and I'm losing marbles by the second.
My absolute pleasure. Have you, from your experience, seen any evidence that current treatments influence the course of Parkinson's?
The answer to that's no, I haven't seen it. It doesn't mean it doesn't exist.
Perfect. And then the second part of it, is there any indication that novel drugs or these kind of new drugs that we're kind of hearing about will actually achieve this? What's our hope factor, shall we say?
Well, my hope lies with researchers like Dr. Alistair Noyce that we saw in the first session, who's doing his PREDICT PD study, and trying to ascertain who in the population is likely to develop Parkinson's disease. Because unless we can target people in the prodromal stage before diagnosis or soon after diagnosis, I don't think we're going to be able to affect the course of the disease. I think there is always hope for novel drugs under trial now and coming onto the market. And I think that having hope is something that's very important for any of us with the condition to hold on to. So I have hope.
Fabulous, thank you. And Simon, what's your take on that? What do you think of the novel drugs that are coming through in terms of their possibility of being, changing the course of the disease?
So an important part of my daily job is to manage expectations, and these sorts of questions, there's obviously, you don't want to be raising expectations too high. But I think when I get asked these sorts of questions in public presentations, I often say that within my lifetime, I expect to see disease modification. I expect us to be slowing down progression of Parkinson's. And within my daughter's lifetime, I think we pretty much are knocking it on the head. She's 9 years old, just to put that in context for you. But if you'd asked me the question before 2017, August 2017, I would have said we've seen very little evidence. But until August 2017, we saw the results of the exenatide trial, and this demonstrated a plateauing of motor features over the course of that trial, 48 weeks. So that was a really encouraging result. And it was coming on the back of literally 20 years of research that is centered around the genetics of Parkinson's. Back in 1997, when we first discovered alpha-synuclein, since then, we've discovered all these other pathways, biological pathways such as LRRK2, GBA, and we're discovering the underlying biology of those genetic risk factors, and that's giving us insights into potentially how this disease is progressing, or diseases are progressing. And I think going forward, if we have molecules that can target all of these different pathways, and we can provide combinations of these therapies to people with Parkinson's, I'm quietly optimistic that we will start to see slowing of progression. It's a complicated question, though, because most people most researchers think only in terms of motor conditions, and a lot of people with Parkinson's are more focused on non-motor features. So I think we need to be careful in how we answer it.
One thing, if I may, one thing we do need to be realistic about is time. Most of us with Parkinson's would like a cure, or to be able to roll back the disease yesterday, and it's not going to happen. Research takes a lot of time, as we know. It's extremely expensive. Neuroscientists have to be able to replicate their results. And it's important for everybody's safety that this is done carefully and scientifically. But I mean, for 200 odd years since Dr. James Parkinson first identified the disease, nothing much happened. And then 60 years ago, we got levodopa. If it takes another 30, 40 years, so be it. But I think the speed at which we're getting positive research results coming through, and hopefully eventually into the clinic, I think there is optimism. But we do need to be realistic. These things don't happen quickly.
90% of the research that's been done on Parkinson's in terms of publications has occurred in the last 20 years. 90% of it.
Yeah.
Which is huge when you put it into the scale of all of the research that's being done out there, not just in the field of Parkinson's, but actually clinically across all condition areas. And especially because we've seen that uptick, COVID came along, that's absolutely changed the way that, research is done, and actually the collaboration and information sharing. So this has been a really, really positive, I suppose, move forward. Thank you so much for your time today. I'm sorry we didn't manage to answer all of the Q&A questions that we had through. There were lots, and it was really positive. There is the extra reading from this session. Please do go and read it. The information that was shared, of course, was a lot. If you would like to know more, do go and follow the links. There's the Hope List, which actually Kevin, who's not in the live Q&A, is behind. Also, you can see what's going on at Cure Parkinson's, which is where Simon is involved, and Paula is heavily involved with us in terms of our research interest groups and our development team. But do go and have a look if you want to take part in research at our Take Part Hub and on our information page. There is so much more, but I hope you enjoy the rest of this conference. I just want to say a massive thank you to Simon and Paula and all of you for sending in your questions. So thank you very much.
Bye. Thanks, Amelia.
Thank you.
Thank you.